Well, I have 12 o'clock, and so I think if it's okay with everybody, we'll get started. So I'm Ann Spolarich. I am at ASDO, and I think I know most of you who are joining us live today. And for those of you who I have not met or who will be watching this recording later, thanks for choosing to spend what I hope will be not only today's hour, but also next week. This is a topic where I was actually telling Deb that I probably need about 10 hours to cover clinical trials because as I've been thinking about this topic and preparing for today, it made me flash back and realize that my very first involvement in a clinical trial was in 1985. So I have been involved in more clinical trials than I can think of, and I've learned a lot of lessons along the way and had some hard lessons. Those are always often the best ones that we learn from when things go wrong, and how we can plan better for the next time. So over the course of today and next week, I'll share with you some of those lessons that I've learned and incorporate some examples into our discussions. So I like to keep things informal. So, I always love to hear from people. We'll have time to chat. But if you're not comfortable asking a question, or you think of something a couple of days from now, please feel free to shoot me an email and I'll get back to you just as soon as I can. So I'm going to go ahead and share my screen with everybody. Can everybody see that? Yes? Hello?
Yes.
Okay, great. Thanks, Kurt. So I'm going to just start this from the beginning. So to start with, clinical trials have a lot of moving parts. And sometimes I think, appropriately focus a lot on the design and the proposal and the content, but there's actually an equal amount of work, if not, I dare say more, in the administrative side of a clinical trial. There's a lot of components. There's the human subjects, there's the contracts, there's a grant, maybe if you've applied for a grant to support this. There's maybe working with the sponsor. There's all of the safety components, the security components of the data. How are you going to report it? How are you going to think about it in terms of your analysis? How can you best answer your questions? And what are all the T's that have to be crossed and I's that have to be dotted for the university in terms of contracts and follow-up? So I've had a lot to kind of think about to put together for you, and I had to come up with a pretty good plan. So here's what I think we're going to do in terms of spending time, because again, best laid plans, there's a lot of material to go through. So today I thought we'd just do a nice little review of experimental design, because a clinical trial is the classic example of an experimental design. And we'll talk about the parts of the project plan. So hopefully you all received the email, because you have registered for this event, I actually gave you a sample template. This is what I actually use myself when I think through a final project plan. Now, before I even get to this document, I'm going to have many iterations on paper, in a Word doc, where I'm thinking about who's my sample, what are the questions I'm going to ask, how am I going to measure it, and it may go through many, many, many iterations. But when I use this document is where I put it all in place, and it reminds me and prompts me about what kind of documentation or supportive information do I need for each of the components of the proposal. So I share this with you freely. I hope everybody uses it, and we'll look at it a little bit closer together today. So I've actually given you some sample language that I use in there, and then in the blue font where you would fill in the information about your own study. And I have spoken with Gayla about about this, I'm happy to share this template for anybody to use in the university, for our university to adopt, because the better we can hone down on the process and for consistency, the better it's going to be for everybody. And I'm totally open to your suggestions for how can we improve this and make it even better. So let's all consider it a working draft. So then next week, I want to kind of switch gears and talk about all of the administrative oversight that goes into managing a clinical trial, from working with our sponsored programs division, making sure all of our contracts are in place. We'll talk a lot about that process. I'll also review our university policies as they relate to research and how some of them will directly influence you in a clinical trial. For example, are you going to give gift cards as an incentive? How do you manage that process? There actually is a process in place for that, and what's the paperwork that goes along with that? We'll talk about some of the IRB implications, and we have some of our IRB folks here joining us today, and that's actually a whole topic in and of itself, but we'll talk about where to find the information, what kind of information needs to be filled out, what are some of the things you need to be thinking about, and then just kind of your general overall management, from the people to the paperwork to the process to the scheduling to all of those kinds of things. Then I'm going to share with you what I like to call my rules to live by. These are the rules that I put in place for myself as the PI and they're really based on lessons learned over the years. And I think of these rules to live by as guidance rules to help you ensure that you'll have a successful trial. And what I mean is that you'll be able to implement that trial and manage everything with the least amount of problems and aggravation so that you actually have a really good experience. So I hope that everybody will plan to join me again next week when we kind of talk about that other whole side to managing a clinical trial. So today, I thought we'll focus on what are the elements of experimental designs, how do we describe an intervention, and then what are our outcomes and how do we define them, and then how do we pick the metrics, right, the measures that we're going to use so that we can actually look at our outcomes in a meaningful way. How do we select appropriate controls for the studies that we're going to do? When is blinding useful in clinical trials? How do we go about randomization, random assignment? And there's different ways that we can look at that, and you have to really think about which strategy is appropriate for the kind of question that you're asking. We'll talk from the 30,000 foot view today about some important ethical considerations to think about when you're planning your trial. And again, then next week, we'll kind of get down a little bit more into the weeds when we talk about the IRB application and protection of human subjects. And then we'll talk through this document today that I shared with you as the working tool for the project plan. So I hope that sounds like a good strategy for everybody today. So clinical trials, as we all know, involve a group of human subjects. We pick them based upon the kind of problem that we want to study, and we're going to compare them. We're going to put them into groups. We're going to have an intervention group. We're going to have a control group. Sometimes we have more than one intervention group. We may have multiple arms, multiple groups. Kind of depends on the question that you're asking. But the intent of any clinical trial is essentially the same, where you want to look at, does the exposure / the intervention cause the outcome? So an experimental study is always grounded in trying to assess causality, right? So the other really important consideration here is the experiment ethically justifiable, right? We want to make sure that the question that we're asking and what we're going to ask people to do, or what we're going to expose those people to, is really ethically appropriate. And we'll talk more about that in a few minutes. So everybody knows that randomized controlled clinical trials are considered the gold standard. And in this era of evidence-based practice, we really look to randomized controlled trials as important sources of evidence to guide our decision-making with our patients. Now, little sidebar here. One of the challenges with our literature today is that it tends to be inherently biased. And that's because typically when you're reading clinical trials, you always hear about, the good studies and what works, and it's kind of slanted a little bit. It's really hard to find a study to say, wow, this was a total blowout, it was an epic fail, we tried this, it didn't work, nobody benefited from that. It's hard to find the other side to the story in the literature. So you have to be mindful of the literature around the topic of interest that you're selecting, and use that to help to guide you. Where are there gaps in the literature that a new study could meaningfully fill? And what is the part of the story that you're hoping your clinical trial will tell? So as the investigator, when you're planning a clinical trial, the advantage you have as the investigator is that you actually know a lot about the intervention, the exposure that we're going to use with the subjects in the study. You know what the dose is going to be, the duration that someone's going to be using something or exposed to something, and how often are they going to be exposed to that. Those are known variables, right, in this discussion. And the beauty of that is that you can compare then the health status of the person before and after the exposure. And that allows us to have a better sense for whether the exposure actually caused the outcome in question. And this design, of course, is the typical one that we use in health sciences. And of course, as I said, it's really kind of the hallmark type of study design. Um, and then, of course, in the hierarchy of evidence, then we look at things like systematic reviews and meta-analyses, where you look at the results of multiple clinical trials and you kind of synthesize the findings into one large outcome that helps to guide practice. And that's what's often used in clinical practice guidelines. So in a randomized controlled clinical trial, remember, you have different arms. So you're going to have people who get randomly assigned to an intervention group, and that's sometimes called the active group. There's different names and terminology that you see thrown around with clinical trials. And sometimes you may have more than one interaction, right? So maybe we just did a big study here on campus with tooth whitening, where the intervention was tooth whitening, but we had different dosages, different regimens, and different lengths of times of exposure. We actually had 5 arms in that study, and they were all active intervention groups. And then we have another group of people who are the control group. They're the comparison group. And we'll talk more about the different types of designs and the different types of controls that you use based upon the kinds of questions you're asking. And of course, all the participants in both or all groups get followed forward in time. And at the end, you assess who has a favorable outcome and an unfavorable outcomes in both groups, right? In both the intervention group and in both the control group. So here's the typical framework for an experimental study, right? So a person gets randomly assigned to the intervention or to the control group, and we want to know, right, what were the outcomes in both groups, and did the individual meet criteria for a favorable outcome? Now, I'll tell you, sometimes in planning a clinical trial, you pick a certain condition that you want to study, and you have to really define, what does that condition mean? So if we say, I want to study diabetes, well, what does that mean? It's such a huge topic, right? So are we talking about people with type 1 diabetes, type 2 diabetes? Do they have both? Do they use certain medications? What's the extent of severity? Where are they in the course of their disease? There's a lot of information just about the people who you could select to be in your study. So you want to make sure that you have a pretty tightly defined set of parameters about the people who will comprise both of these groups, because if you have a lot of variability within each group, it really dilutes what you get out of this study. I mean, if somebody doesn't have a favorable outcome despite getting an intervention that was supposed to help, well, could it be that the person's disease state was in such an uncontrolled state that no intervention was really going to make a meaningful difference for that group. So really understanding who the people are and the condition you're studying and the intervention that you're studying is very important, because you want to have a certain level of confidence that the outcomes that you're seeing are in fact due to the intervention and not from other kinds of things that we call confounding variables. So when we think about this, there's again some important things for you as the person designing the study to think about. I have to define what the intervention is. What does it really mean? It has to be something that everybody on your research team really understands, and most importantly, that the people who are in the study understand. What is it? How will you randomize participants to the groups? What type of control should we use for comparison? And then, as I started to say, what would be something I would consider to be a favorable outcome? And it should be something that's so well defined that it's pretty tight and pretty clean, and that whatever measures or tools that you use to assess that clinical condition are really the best choices for helping you determine what the outcome is. And then, of course, you also have to think about the ethical challenges of exposing people to certain kinds of interventions. So when we start by describing the intervention, we think is, what is it going to be? Well, is it going to be an educational intervention? Am I going to train patients to do something? And then am I going to look at their behaviors before and after? Am I going to ask them to take a drug? And I want to see what that drug does in terms of maybe effect on their red blood cell count, or on their level of glycemic control if that patient's diabetic. Where and how will the participants receive the intervention? Do they have to come and be right in front of me? Do they have to actually see me in order to get it, meaning I'm actually going to administer the intervention? Or is it something that people can do at home on their own? And then as the investigator, I have to think about, how can I tighten that up so that I can do everything in my power to make sure the person uses the intervention as intended? So we do a lot of mouthwash studies and oral care product studies here in the dental school. So if I'm going to ask you to go home and I say, I want you to rinse with this mouthwash twice a day, well, I didn't really give you a whole lot of information. What I have to think about is, I want you to use 15 milliliters of this mouthwash. Here's the line in the cap, that's the demarcation that I want you to use to measure it each time. I want you to rinse and hold that in your mouth for 15 seconds. I want you to spit it out. I don't want you to rinse it out. I don't want you to eat or drink anything for 30 minutes after you do that. That's a very different description of the intervention compared to, here it is, go ahead and do it and have at it and let's see what happens, right? So when you have people do interventions for you on their own, there's a lot of things that come into play. And the biggest one is compliance, right? Because the thing about clinical trials is we use human subjects, and people are people, and they have their own opinions and their own attitudes and their own behaviors, and they tend to do their own thing no matter how well informed and instructed they are. So you always have to think about, if somebody is going to do something on their own, you must figure out a way to assess compliance, because it's going to directly influence the outcome of your study. If I have to do something to them, or they have to do something in front of me, then I have to think about, well, where am I going to do that? When am I going to do it? Does the time of day matter? Do other behaviors that the person has potentially confound what I'm going to be asking them to do? So is timing a really important variable for us? So when, how often, and for what durations will they get the intervention? You know, do they have to do it every day for 6 weeks, or 5 times a day for just 1 day? You know, what is it going to be? Because the more often somebody has to do something, you have to realize the more likely it is the person won't be as compliant. The more complicated the intervention is, with more complicated instructions, the more likely it is it's going to influence compliance. So these are all things you have to think about. And here's one of my rules to live by. If it isn't something that I'm willing to do myself, can I really ask somebody else to do this? So like, I have a rule as a dental professional. I can't ask anybody to put something in their mouth that I won't put into my own mouth. So if I'm not willing to brush with that, rinse with it, swish with it, whatever it's going to be, I really don't feel confident asking somebody else to do the same thing. So think about in your own discipline what some of those rules might be for you, and how it is that those activities will affect somebody's compliance. So most of the experimental studies that we do are typically designed as what we call superiority trials, where the intent is that the assumption is the intervention is going to be better than the control. So if I switch you over to this product, you're going to have a better clinical outcome than whatever you're using now, or than not doing anything at all. Now, we have some other kinds of designs to think about. Sometimes we have studies that are called non-inferiority trials, where what you want to show is that this new intervention isn't worse than the control, right? So the person wouldn't be sacrificing anything by giving this particular intervention a shot. And an equivalence trial is where you intend or hope that the intervention will be equal to the control. So we're doing a study right now with patients undergoing head and neck cancer, and they apply a very high dose fluoride to prevent dental caries, which is a known and rapidly developing outcome in patients who get radiation for head and neck cancer. And the standard, which took us about 50 years to get into place as the standard, is to have patients put the fluoride into what we call a custom tray, where we take mold of your teeth, we make trays that kind of look like a bite guard, and the patient wears it in their mouth for 5 to 10 minutes. Now, here's the problem. If you're going through cancer treatment, you're often nauseous. You know, your mouth is sore, you're feeling crummy. And we know that compliance with the trays is not good, especially during certain times of the treatment period. So we said, well, what would happen if people brushed on the high potency fluoride instead? Because people are used to brushing their teeth. And even if you feel crummy, you're still most likely going to brush your teeth. And what we knew anecdotally from working with these patients in the past was that a lot of patients had gotten trays and they said, you know what, for like the first 3 months of my treatment, I just brushed it on instead. So we knew people were already doing this. So we're actually doing an equivalence trial right now with the hopes of being able to say, hey, if you're not feeling so well, or if the course of your cancer treatment is more difficult for you versus somebody else, that brushing will still get you a good therapeutic effect. And we also are going into this with the assumption that compliance will be better with the brush group, because it's something people do anyway. And it's quite interesting to see what's happening. And I can tell you, our data are sort of trending just as we had anticipated. So when you think about, well, what is your intent and what do you hope to be able to tell as your story at the end of the trial, is thinking a little bit about how you want to position your outcome. So then we get back to this issue of how do we select an appropriate control. So the most common is with a placebo. Sometimes it's called sham. So we're all familiar with the placebo effect. It's quite interesting when you look to the literature. We know there are certain populations that have what we call high placebo responders within those populations, where people will respond to drug therapy even if you're giving them a sugar pill. There's some interesting explanations for that, which are a little bit beyond the scope of what I had planned to go into today. But you do always have to be aware of the fact that you will likely have placebo responders in your group. And depending upon the phenomenon that you're studying, the percentage of those placebo responders may be higher. So for many years, I was involved in studies that looked at pain and trying to do quantitative ways to assess pain by looking at things like blood pressure and different chemical biomarkers in the body, electro activity in muscles, things like that. But we also know that pain is quite closely tied to things like depression and anxiety, depending on the chronicity and severity of the pain. And we know that in people who take antidepressants, the percentage of placebo responders may be as high as 60% versus in another population where you're doing a drug study where the placebo effect may only be apparent in up to 30%. So it really depends on who your population is. So you know, there are going to be people who will report feeling better and behave differently because they're in a study, and more on that in just a minute. So the beauty, though, of having a placebo-controlled study is that it allows the effect of the active therapy to be examined separately from what you see in the change in health status that a lot of people will experience or report simply because they're participating in a study and or because they're getting you know, to take part in an activity or receiving some type of an intervention. There's some really interesting studies where people respond better to pills that are different colors compared to using like a white pill, right? Same shape, same size. The only thing that's different is the color. And if they're all sugar pills, you'll have people who will respond differently just based on their own perceptions. I work in geriatrics, for those of you who know me personally, and in special care. And, you know, one of the things I've found over the years in doing studies with older adults is that a lot of older adults who are in the control group seem to do better. And part of it is, you know, being in the study gives them a purpose. They get to interact with people. It's important for them. They know they're contributing to something. It's a really neat phenomenon to kind of look at by itself on the side. So a couple things to be thinking about. You know, it's not always ethical to use a placebo if we know that an effective therapy is already available. In the early years of my career, I worked in hospitals and I started out working in oncology. And boy, I lived this statement because, you know, it's pretty hard to withhold therapy from somebody who has a life-threatening illness, especially if we know there are therapies that are effective for that person. And this is where we often will have, you know, different treatment arms, but we compare a new intervention to the existing gold standard intervention instead of having a control. So you have to really be able to justify the control. Now, so just what I was talking about is sometimes you have like an active control where a person's already getting like the usual custom care or the standard of care, and you're trying to compare something new against that. Now, sometimes we want to know if we add a therapy onto the existing standard of care, do you get, you know, like an additive or a potentiated effect, where in that kind of a design, you know, everybody gets the standard of care, and then you have an arm that adds the new therapy to that. So they're doing two, right? So we have different ways that we can think about these designs based upon the question that you're asking. So if you look on this chart, you see on the left the different types of controls with the different arms of the active intervention. So of course, the top one is that really classic placebo, which is inactive compared against the new active intervention. So that might be something that you're testing an active ingredient, a therapy, a substance, a procedure, whatever it might be. The second one is when you have the standard of care, we just talked about this, against the new therapy, or then that additive one, the standard of care and the new therapy. Dose response studies are what we typically see in pharmaceutical research, where you're looking at a medication, some kind of an intervention, and some kind of a dosage pattern and some kind of a duration pattern. And again, lots of these studies often will have multiple arms. So like in our whitening study, everybody was using hydrogen peroxide to whiten their teeth, but we had 5 groups. Everybody was an active group. We had no placebo control group in that study, but we were looking at different concentrations, different routes of application for different durations of time. And we were trying to compare to see which one produced the best outcome, which was the number of shades of improvement of lightening the teeth. So you can also have no intervention to the new intervention, or I've done a lot of these kinds of studies too, where each person serves as their own control, right? So you have the person's health status before and after the intervention. Now, you can also do this in what's called a crossover design, where let's say, I'm trying to come up with something other than a dental example. Let's say we want to try, I don't know, a new exercise, a new weightlifting regimen, and we want to look at that in terms of effect of upper arm strength. I'm making this up off the fly. So we'll have, regimen A and we'll have regimen B. And then we're all going to do some kind of a strength testing measure for the outcome. So in this case, what we do is everybody's randomly assigned. You get randomly assigned to start with A or randomly assigned to start with B. And everybody does their regimen for 2 weeks, and then you have your test, right, to test the outcome. Then there's usually a period of time. We often call this the washout period, and then we crisscross you and you switch over, you do the other intervention for 2 weeks, and you do the test, and then you compare them, right? So each person serves as their own control. So earlier I was talking about the fact that, some people change their behaviors and they do better because they're in a study. This is known as the Hawthorne effect, and it's a type of bias that you see a lot in clinical trials, or the risk is there for the Hawthorne effect, where participants will change their behavior for the better because they know they're in a study. Now, I find that this happens more, or my personal experience has been, when people get randomized to an intervention versus their normal routine. So let's say that our study is to look at the effect of dietary choices on our level of glycemic control, and I get assigned to my usual routine. In the intervention group, those people get to have a session with a dietary counselor who's going to train them about food choices and how those foods relate to glycemic control. But, because I had to sign a consent form, I know I could be assigned to the intervention group or to my regular routine. So obviously I don't get called by the dietary counselor. So I figure out I'm in the normal routine group. And so what happens? I know that in a couple of weeks, they're going to test my blood sugar to see what happens. So I know what I'm supposed to do. So I start making better food choices, right? Because I'm in a study and I know somebody's going to be looking at me. That's a really good example of the Hawthorne effect. So it makes it really hard to accurately measure the impact of the intervention itself. So you always have to think about that and think about what could you do design-wise to maybe try to reduce that risk. So when we think about some other considerations, we always, of course, have to think about the ethical concerns about the appropriateness of putting people into an intervention group where, the intervention is somewhat risky, and then also not assigning people to a potentially life-threatening, life-saving intervention. So, you have ethical concerns on both sides of the story. And of course, you're going to have to talk about all of those things in your IRB application. You're going to have to justify what you're doing and what kinds of things can you do to mitigate some of those risks. And is it justifiable? I mean, does the end justify the means? That's ultimately where the bottom line falls down. Time itself is often a confounding variable, especially if you're working with people who are very sick, because the natural course of their disease progression can really confound your results. So we can't control time, right? So you may see declining health status that, in my experience, is the more common trend, or improvements in health status if you're talking about maybe an infection and you're waiting for somebody's immune response to help them get better. So, what are you going to study, and is timing really critical when you're going to do the intervention, because the natural time course of the problem in general is going to be something that can influence your results. And always recognize too that studies can be stopped early if an intervention is clearly working or it's clearly causing harm. And you often hear a lot about these kinds of studies in the media while they're in progress. And this is something that happens quite a lot in pharmaceutical research. So everybody has heard about the term blinding. It's sometimes called masking, and this is what we do to minimize what we call information bias. And it's usually only possible when all the participants are assigned to a similar exposure. And a single blind study means that the participants are unaware of their status. So I'm going to ask people to test a new mouthwash, but I know which mouthwash is the active one and which is the placebo one. The person does not. So everybody has the same shape bottle, the same color mouthwash. It tastes the same. The only thing that's different is the active ingredient. And the person doesn't know which one they're getting, but I do. If it's a double-blind study, neither one of us knows what they're getting. And I see my friend Mark Schlossman is on this call. Mark is my partner in crime here in clinical research at ASDO, and we've done a lot of these kinds of studies where actually the product company will label the bottles for us. So even we don't know, they'll call them. We did one study where it was like product G and product H, so you couldn't get into that assumption of A and B, like we have, certain values that we always have inherent biases with. There's actually, by the way, something called a triple blind study too, where the people who are analyzing the outcomes don't know the intervention either. So just so you know, it's always good when you can incorporate this into a clinical trial. And of course, our gold standard is randomized controlled clinical trial, preferably double blinded. So if it's not possible, you want to try to figure out some other objective measure that's not subjective when you're trying to look at the outcome. So like a laboratory test, or you can use some kind of a device to measure the outcome where your own subjective assessment gets taken out of the equation. So on this slide are some schemes for randomization. So how do you go about randomizing the subjects in your study? So lots of us use what we call simple randomization. You can see that on the left-hand side of this schematic, which you think about as the coin toss. You get into this group or that group. And typically we use something called a random number generator to help us figure out the pattern. In other words, we don't actually come up with the pattern ourselves. You can, access these. There's software programs from this. You can find them on the internet, and there are some challenges with some of them, but the point is you use a program to generate the order in which subjects get assigned. Now, block assignment is used if you have groups of people who will be assigned to a group. So, maybe you're doing an intervention in a community with some kind of a public health measure. And so you would actually say, well, everybody who lives in Mesa is going to be compared to the group of people who live in Gilbert, or all the kids who are, at Mesa Middle School on the soccer team with concussion will be compared, to the Gilbert Middle School kids who are on the soccer team with concussion, right? So you're looking at groups of people who will be compared, so as opposed to individuals. And then sometimes we do what's called a stratified randomization, where you have subgroups of people. So if you want to make sure you have good representation by sex or by age, these are some of the more common variables. And that's really helpful when simple randomization won't result in enough members of certain subgroups to be randomized to each group. So there's advantages and disadvantages to each of these approaches to randomization, and I would encourage you to find like a good textbook or a good article to read about that to help guide your decision making with randomization. All right, so I wanted to just talk for a few minutes about some of the 30,000-foot view when we look at the ethics of a clinical trial. And I'm sure our IRB folks are loving this part, because this is where they live and breathe. And again, we'll talk a little bit more about this in a deeper dive next week. But we have certain concepts that I think are all-encompassing when we think about the protection of human subjects. So equipoise is when we talk about the fact that experimental research should be conducted only when there's genuine uncertainty about which treatment will work better. The concept of distributive justice means that the population source, right? Where are you going to pull your potential subjects from? Has to be an appropriate one for your study. And that it's also really important to recognize that your study won't exploit low resource individuals who are unlikely to have continued access to a therapy if it's found to be successful. So we have to think about the ethical impact of, hey, you get to use this intervention that does all these wonderful things for you, and oh, well, it's 6 weeks later and the study's over, and you know, you're out of luck. Right, so you have to really, really think about the impact of that on those individuals. Respect for persons is a fundamental hallmark of human subjects protection, where all of the participants have to volunteer without being unduly influenced by the prospect of being compensated for their participation. And of course, when we look at the IRB application, we have a whole list of people who we consider to be vulnerable individuals, vulnerable populations, where we worry about this issue of coercion. Are they a captive audience, and we can unduly influence them so they volunteer, but it's more like they're voluntold, and that's not a good thing, right? And that's why we have protections in place for those kinds of populations. More on that later. But we also need to make sure that people really understand what it means to be in a research project, including the fact that they could be assigned to the control group instead of to the new intervention. So people really have to be quite clear, and this is where I personally like to use one of my rules to live by, is what's called the teach back technique. That person, when I'm talking with them and consenting them into the study, that they really understand what the parameters are, and the fact they might get put into a group that they don't really like, right? So let me give you an example. A couple years ago, we had a student who was very involved and used to train dogs for pet therapy. And she used to go into hospitals, like we're all familiar with pet therapy. It's such a neat intervention. And she was very interested in doing this in our dental clinic with people with severe dental anxiety. And of course, some of the people in our advanced care center have intellectual and developmental disabilities. And we were actually looking at clinical measures, salivary cortisol secretion, blood pressure, pulse rate, as some of the measures we were doing to look at the effect of the pet therapy intervention on these clinical outcomes, with the assumption that these markers go down as stress comes down, and that the intervention of being with the dog would help to reduce anxiety. But of course, people would be randomly assigned to the dog group or to treatment as usual. And so the ethical dilemma, especially for people with intellectual and developmental disabilities, but for all people, was that we knew that people would feel really cheated if they didn't get assigned to the dog group, right? Everybody wants to be with the beautiful golden retriever. And, so there was a risk that people would actually develop more anxiety when they saw somebody down the hall with the dog in the room and they didn't get that intervention. It was going to actually confound our results. So when we planned that study, we actually planned for the people in the treatment as usual group, the control group, to collect all their data first, and then we offered time with the dog at the end of the appointment as a way to help to promote some fairness of time with the dog. So that's a really broad overview of that study. There were many moving parts to that, but it's just kind of an example of what we're talking about here, right? All right, so the other really important components here are really, aiming for beneficence, where we're doing good, right? The intent of the study is that we're trying to help somebody and we're not trying to cause harm to the individual. So this is where we look at the balance of benefits and risks of participating in the study. And again, this idea of what is the right type of control to use in this situation. Now, a lot of studies, especially if you're testing a product or you're testing a drug or even a certain medical device or an intervention, will pose the likelihood that an adverse event could take place. So you have to put a monitoring system in place in a clinical trial to watch for that. So it might be as simple as Having a form that you fill out each time you examine a person in a subject, and sequential study visits for, was there an adverse event, and then how do we document it, and what do we do about that? And next week, we're going to talk all about, if an adverse event occurs, how do we report it? Who gets notified about that? The IRB, the study sponsor, and how is that managed? And we're going to talk about something called a data safety monitoring plan, which for certain kinds of studies have to be put in place as part of your quality assurance part of the study. So you also want to think about conditions under which an experiment might be discontinued early. Do you have a whole lot of people who are having a significant adverse event with testing this product? And maybe that means that the study has to be halted. So in this case, where the exposure would prove to really be promoting the risk, and then also thinking about the flip side of that, where the new intervention is so beneficial that keeping it from the people in the control group is actually unethical. Okay, so rocking and rolling here. So again, we'll kind of talk more about this next week, but just to give you again a little bit of an overview, you really need to be thinking about all of these things because you're going to have to address them in the IRB application. So, all of the people who are in your study, the risks and benefits, what are the components of your informed consent process, and you're actually going to have to develop your consent forms, and the IRB will look at your template consent forms that you have written up for your study, and to make sure that you have actually covered all of these points in terms of risk benefit. But you also have to think about, how are we going to protect somebody's confidentiality and their privacy, especially if you are using protected health information in your clinical trial. How are you going to be monitoring for safety, conflicts of interest, the training of the researchers, and all of the ways that you're going to be documenting your data, and then things like adverse events. So these are all things you have to think about anyway in your project design, and they're going to translate into your IRB application. So part of your data analysis plan is also based on some of the specific components of your design. So are you going to look at ideal world efficacy, where that study is super tightly controlled, like so many clinical trials are that you read? Or are we looking for real world efficacy, where you want to maybe have people do something like they would typically use a product, or take a medicine, or whatever you're going to ask them to do, what they would typically do in real life. So the approaches to the design are a little bit different, and then that translates into some of the ways we think about analyzing the data. So we have what that ideal world efficacy is called a treatment received approach, where the data analysis will be limited to the participants who were fully compliant with all of the instructions for surrounding the intervention. The treatment assigned approach is sometimes also called the intention to treat approach, where your analysis would include all the people, all the data you got from all the participants, even those who weren't fully compliant with the assigned intervention. Maybe they didn't follow instructions, or they skipped a couple days, or whatever they did. And then you recognize and have to address some of those limitations. So this is a discussion I think you need to have with your biostatistician when you're planning the design of your study, and also thinking about going back to, what are we asking people to do? Because the more complicated it is, the harder it is to control because of the compliance component. So I can tell you, of all the trials I've been involved in, and I've been involved in both, most of the ones that I'm involved in are real world efficacy. And then sometimes, you think people are doing really, really well, and then you might have to make a decision about, well, hey, these 3 people really were outliers, and they really didn't follow the rules. And how are you going to address that when you write up your study? So in typically things like cost benefit analysis types of studies, you get into some other kinds of concepts related to efficacy where you figure out the number needed to treat and the number needed to harm. So what is the impact of an exposure on the likelihood of having either a favorable or an unfavorable outcome? So again, with efficacy, you have to look at, you know, favorable and unfavorable outcomes in both groups, intervention and control. But here we're really looking at the proportion of people in the control group who have an unfavorable outcome, who we actually could have expected would have had a favorable outcome had they actually been in the treatment arm instead. So the more efficacious an intervention is, the more likely that it is, quote, successful. So you've probably heard these terms before, the number needed to treat, which is the expected number of people who will have to receive the treatment to prevent an unfavorable outcome in one person, right? So the small number needed to treat, it represents a more favorable intervention. And then the number needed to harm would be the number of people who would have to receive the treatment in order to expect that one of them would have a certain type of adverse outcome. So the larger the number needed to treat, the more effective it is. So again, a lot of these kinds of considerations are based in cost effectiveness studies, and I wish we had more time to go through that, but there's a little more info on these slides, and you can certainly read more about it in a good resource. So what did that study find? My number needed to confuse. So that's a good little kind of ending point for that. So I'm going to stop sharing this particular slide deck, and hopefully I can share with you. Sorry, let me go back here. I want to share this document with you. This is the one that we emailed to you, which is the template. And can everybody see that all right? So I actually want you to think about this as you work through it. And as I said, a lot of this is some sample language in the document that I use. And the first part is kind of the bigger overall picture, just a couple of paragraphs, give me the 30,000 foot view. This is a randomized controlled double-blinded clinical trial, and we're going to look at changes in this in people who have that, who choose to use this product. And it just kind of gives you the little overview, if you will, of the design. And then as you work through the different components of this document, you can actually, I don't know if it's going to let me scroll. Yeah. So here's where you figure out how many subjects do I need. And the way you do that is you meet with your biostatistician after you think about what are the arms of the study, what are we going to ask people to do, what are the variables we're going to measure, and you do what's called a power analysis, right? So I'm going to talk next week. Biostatisticians are such critical members of our team. I believe in meeting with them very, very early and often, right? They should be critical people to help you in the design of your study, because there's nothing worse than at the end of the study, you have a bunch of data, you don't know what to do with it, and we expect our biostatisticians to leap tall buildings with a single bound. And they are all fantastic at our university, but we don't ever want to put them in that kind of a position, right? So do this early to figure out some of that information. How long is your study going to take? Here in investigational products, if this is relevant to your clinical trial, this is where you want to talk about them. You would want to share, are the products FDA approved? Is it a medical device that's been FDA approved, or has it just been cleared? What's the status of it, or it's new? Nobody's used it before, so if somebody's going to take it, are they going to grow a third eyeball? You have to really be transparent about this kind of information. So this is where you want to write about it. You want to talk about your study materials. How are they tested? How are they packaged? How much do you need? Now, I actually do this with math, right? So if I'm going to ask somebody to use a product and they need to use this many milliliters twice a day for 10 days, then I can calculate out the amount the person needs and how does it come packaged. So then I know I'm going to need 10 bottles per person times so many weeks, and this is what I expect to see. And you want to show the math, and you also want to compensate for what we call over enrollment or dropout, right? Because some people will drop out along the way, and so you always want to have some extra on hand. Your inclusion and exclusion criteria, they should be super specific. You know, what are the rules to be selected as a participant in the study? You have to have X, Y, and Z conditions. You have to be of this age. You have to be free of that. You've got to be ambulatory. You have to have brown hair, you know, whatever it is, right? And then your exclusion criteria would be all of the things that would, you know, automatically knock somebody out of the running. So I can give you examples of these if you need help doing that. You want to talk about your screening procedures because, you know, sometimes you have to look at people and figure out, you know, well, they look pretty good on paper, but maybe I need to interview them and go through these questions with them again. Maybe I need people who have brown hair and you show up and you have red hair. You know, I need to see you. I need to figure it out. If I'm going to do a study in people with diabetes, how do I know you have diabetes? You know, do I need medical confirmation? Do I need to see a copy of a lab test? Do I expect you to go have a lab test, right, as a part of that? So here's some of the things, some things to think about in how you screen somebody and how you determine their eligibility. And then you want to describe your process of enrollment. And this is where we talk about the informed consent process, right? Then again, your randomization. What's the process that you use? And then the next section is all about your procedures. Most of the time you think about this as the methods, but you're really step by step by step, and there's a lot of information here. So I'll ask everybody to please kind of read through this, and then we can ask questions and talk about that when we're together next time. The next section of the summary of the visit is really what I would ask you to do is like an itemization, like a flow chart or a bullet list. And many times you'll actually use that diagram in your consent form. So it should be something that everybody understands. First, I'm going to have my picture taken, and then I'm going to have my strength testing done, and then I'm going to have to do 10 jumping jacks, and then I'm going to have to, you know, whatever, right? So bullet it out so everybody understands what is being expected. And it also, by the way, makes a great checklist. We're going to talk about checklists next week to kind of track compliance, and that you as the investigator are doing all the procedures that you have planned to do in each of those study visits. And then the last part of this really talks about how will you manage people who withdraw from the study, who are considered to be off study because they, you know, have early termination of the study. They have completed all their follow-up studies, and so they're actually done with the study. Anybody who's withdrawn consent, so what your process is there, what your process actually is for removing people and how you're going to document that, and the responsibility of the PI. Your outcomes of the study, including specific time points, would be listed here. You might want to include all your variables in there, your hypotheses, your statistical analysis plan, and then your data management plan. You know, how am I going to capture it? Where am I going to store it? How long do I have to keep it? And by the way, this is all part of your IRB application too, right? Security of your data, and then your record keeping plan. And then sometimes, depending on who you're working with, you might be asked to work with an independent study monitor that gets hired by the sponsor. And so what are you going to do in preparation for your monitoring visits? Define the parameters of that. And then last but not least, your timeline. So this was, you know, a lot of information, but you can see why I hope having a template document will be very helpful to you as you try to get all your thoughts together and in a very organized way. All right, so let's see if I can come back to you. Okay, somehow I have messed myself up here. Stop share. There we go, I'm back. All right, so we have about 2 minutes. I told you it was going to be a lot. Does anybody have any thoughts right at this moment you want to share. And if those of you need to run off to your next commitment, please feel free to do so. And thanks, and I'll see you next week. Any thoughts? You're still like reeling from all of this information. Dr. Schlossman, did we miss anything? Dr. Bay?
That was pretty comprehensive. A lot of things to fill in under each of those bullets.
Yeah, lots
Massive
of,
amount of information that would require, as you said, many sessions.
yeah, lots of things to think about. Lots of
That
things.
was great. Thank you.
Thanks, Dr. Bay. Anybody else? So just to remind everyone, next week again, we'll talk about some of the administrative aspects of clinical trials, and I'll take you through my rules to live by, and then any questions that you might have. And if you have examples from your own studies, that'd be great. We'd love to hear about those too. So thanks everybody so much. I hope you have a wonderful afternoon, and I'll look forward to seeing you next week. Thanks everybody.